everyone, welcome you all to the Quizzical Doctors. Myself Shordip Nandi, and I will discuss about neuroanatomy. So neuroanatomy is a part of neurology. Neurology is a broad science that discuss with the brain and spinal cord. Among that there are very many disciplines. One is the neuroanatomy, neurophysiology, neuropathology, neuromedicine, and there are many other sub-disciplines also. Now I will discuss about the neuroanatomy, which tells us about the various structures and arrangement of those structures in the nervous system. Now the nervous system, it is divided into two parts. One is the central nervous system and one is the peripheral nervous system. The central nervous system includes brain and spinal cord, whereas the peripheral nervous system has two components. One is the afferent component and another is the efferent component. The afferent component takes the information from different receptors present in the body towards the central nervous system, so it composed of the sensory part. And the efferent part, it carries necessary information from central nervous system to different parts of the body, either muscle or visceral organs. So it is all again divided into somatic neural system and autonomic nervous system. Somatic neural system is voluntary and the information is carried to the skeletal muscle, and the autonomic nervous system is involuntary and it gives supply to the visceral smooth muscle and glands. Visceral smooth muscle and glands. Now coming to the development of brain. So discussing the development of brain, at the fourth week of intrauterine life, we have three brain vesicles. One is prosencephalon, one is mesencephalon, and another is the rhombencephalon. So there are three vesicles: prosencephalon, mesencephalon, and rhombencephalon. In the fifth week of intrauterine life, the prosencephalon matures and divides in two parts. The rostral portion, that is the cranial portion, bulges out and forms the telencephalon, and the caudal portion remain as it is and forms the diencephalon. The mesencephalon do not show any change, whereas the rhombencephalon, it also becomes divided into two parts. One is the metencephalon, another is the myelencephalon. Now coming to the adult derivatives of brain, the prosencephalon matures, the telencephalon matures and gives the formation of the cerebral hemispheres of brain. So here is the formation of cerebral hemisphere. The diencephalon gives rise to the structures, that is the internal brain structures, which are the thalamus, the hypothalamus, subthalamus, epithalamus, and metathalamus. The mesencephalon gradually thickens and narrows down the cavity present within it, and that cavity is named as the cerebral aqueduct. The metencephalon develops into pons. The metencephalon bulges out and develops into pons. And the myelencephalon develops into medulla oblongata. The myelencephalon develops into the medulla oblongata, and the pons and cerebellum is developed from the metencephalon. So let us again discuss. So first we had prosencephalon, we have mesencephalon, and we have rhombencephalon. In fifth week, prosencephalon divides into telencephalon and diencephalon. The mesencephalon remain as it is, and the rhombencephalon develop into cranially the metencephalon and caudally the myelencephalon. Telencephalon gives the adult derivative, the cerebral hemispheres. It is also known as the cerebrum. The diencephalon gives the internal brain structures, that is thalamus, hypothalamus, epithalamus, metathalamus, subthalamus. The mesencephalon gives rise to the midbrain. Metencephalon gives rise to the pons and cerebellum from the metencephalon. Myelencephalon gives rise to medulla oblongata. And the cavity which you can see here is the ventricles. So here is the left ventricle, here is the third ventricle, here is the fourth ventricle. Okay, now let us come to the external features of cerebrum. So cerebrum, it is the largest part of the brain. It consists of two cerebral hemispheres, which is connected by a band of white fiber known as corpus callosum. Now each hemisphere consists of an outer layer of grey matter called as cortex, inner mass of white matter, large masses of grey matter that are embedded in the basal part of white matter, that is present in the basal part of brain, which is called as basal ganglia, because ganglia. Why? Because these are collection of gray matter that is by ganglia, and it is within the basal part of the brain, that's why basal, and a cavity called as lateral ventricle. Now coming to the external features of brain. So for the poles, so here in the front there is the frontal pole, here is the occipital pole, and here is the temporal pole. So these consist of 3 poles. Now coming to the borders. Okay, before discussing the border, let us discuss the lobes of brain, the lobes of cerebral hemisphere. Each hemisphere or the whole of the cerebellum consists of 4 lobes, that is frontal, parietal, temporal, and occipital. Now to demonstrate this lobe, I have to draw a diagram of the brain. So this is the diagram of the brain. So you have to just remember 3 or 4 landmarks to distinguish these lobes. So here at the middle there is a sulcus, very deep sulcus. It is known as central sulcus. Here is another very deep sulcus, which is the lateral sulcus. Here is a notch present at the inferior surface of the brain, which is called as pre-occipital notch. And here is another sulcus, which is termed as parieto-occipital sulcus. Now we have to draw 2 imaginary lines. The first imaginary line joins the parieto-occipital sulcus with the pre-occipital notch, and another secondary line joins the terminal end of the lateral sulcus with the midpoint of the first imaginary line. So here is the first imaginary line, and here is the second imaginary line. So now let us draw the lobes. Anterior to the central sulcus and above the lateral sulcus, there is the frontal lobe. Anterior to central sulcus and superior to lateral sulcus, there is the frontal lobe. Now come to the parietal lobe. Posterior to central sulcus, superior to lateral sulcus and the second imaginary line, and anterior to parieto-occipital sulcus, there lies the parietal lobe. Now coming to the occipital lobe. Posterior to parieto-occipital sulcus and the first imaginary line lies the occipital lobe. Now coming to the lateral sulcus or the temporal lobe. So the part of the brain, cerebrum, that lies inferior to the lateral sulcus and the second imaginary line and anterior to the first imaginary line lies the temporal lobe. So this is the frontal lobe, parietal lobe, temporal lobe, and this is the occipital lobe. Okay. Now based on this, there are some poles. Here is the frontal pole, here is the temporal pole, and here is the occipital pole. Now let us discuss the surfaces of the cerebrum. So discussing the surface, so we have 4 surfaces. One is the superolateral surface. So this is the superolateral surface. If we see the hemisphere from the medial side, then it will look like somewhat like this. Look somewhat like this. So there will be the cross section of the corpus callosum. So this is the superolateral surface, this is the medial surface of brain, cerebrum. And if you see, and there is the inferior surface. So here is the inferior surface. The inferior surface is divided into two surfaces. So whole of it is the inferior surface. It is again divided by 2 surfaces. One is the orbital surface, another is the temporal surface. Why it is named as orbital? Because it just lies over the orbital plate of the frontal bone. That's why it is called as orbital surface. And why it is called as temporal surface? Because it is the part of the temporal lobe, and it is the inferior surface of the temporal lobe. So both of it forms the inferior surface. So there are 3 surfaces. There is the superolateral surface, there is the medial surface where we can see the cut section of the corpus callosum, and there is the inferior surface where we can see another two different surfaces, the orbital and the temporal surface. Now coming to the borders. Discussing the surfaces, there are various borders that divides the surface. One is the superomedial border. Second is the inferolateral border. So superomedial border, it divides or it separates the superolateral surface with the medial surface. So it is present between the superolateral and the medial surface. Inferolateral border, as the name suggests, it is present between superolateral surface and the inferior surface. Other borders are the number 3 superciliary border, number 4 in medial. There are two medial borders. Medial border is again divided into two, that is orbital and occipital. Let us see all these borders in a diagram. Now with the help of a diagram. Now suppose this is the superolateral surface of the brain and I take a coronal section of this cerebral hemisphere. So this will somehow look like this. This will somehow look like this. So here we have this one border which is termed as the superomedial border, which separates the medial surface from superolateral surface. Now here is another border which separates the superolateral surface from the inferior surface. So this border is termed as inferolateral border. Lateral border. Now if we see the brain from the inferior side or the what do I say, or the cerebral hemisphere from the inferior side, so we can see another two borders. So this border is called as superciliary border, third one, and this border is termed as inferolateral border, which is this border only. Here you can also see that the border which divides or which separates the medial surface from the inferior surface is the inferior border, but it is interrupted. So this medial border is called as medial orbital border and it is called as medial occipital border. So medial border is divided in two parts, orbital and occipital. So I think everything is clear right now. Now let us discuss the sulci and gyri of cerebral hemisphere. It is also an external feature of the cerebral hemisphere. Now what is sulci and gyri? The surface layer of the grey matter is the cerebral cortex. It is highly extensive in man, and to accommodate the limited space available within the rigid cranial cavity, the surface of the cerebral hemisphere becomes folded and producing numerous convolutions which are separated by some fissures. So these convolutions which is formed in order to which is formed due to the folding of the cerebral hemisphere is termed as gyri, and the fissures in between these convolutions are called as sulci. So human brain is very extensive, so which leads to folding of cerebral hemisphere to accommodate it inside the rigid cranial cavity, which lead to formation of convolutions, and between the convolution there is formation of fissures, that is gaps. These convolutions or the elevated ends, elevated regions are called as gyri, and the fissures or the gap are called as depressed regions or the sulci. Now let us see with a diagram all these gyri and sulci. You need not to remember every gyri and sulci, at least remember the important ones, and the important ones I will be teaching right now. So here is the superolateral surface of brain. Here is the superolateral surface of the brain. Now the first we will draw some of the important sulci. So the first and the foremost important sulci, which is very deep and almost makes the half of the cerebral hemisphere, is the central sulcus. So this centrally placed sulcus is the central sulcus. Anterior to this there is the precentral sulcus. Posterior to this there is the postcentral sulcus. Okay. In the frontal lobe there is two sulcus again. One is the superior frontal sulcus, one is the inferior frontal sulcus. Now here, which is the largest sulcus or the deepest sulcus of the brain, is the lateral sulcus. So this lateral sulcus has three parts. One is the ascending part, and one is the anterior part, or you may also say this like this, that one is the anterior ascending part and another is the anterior horizontal part, and the part of lateral sulcus that goes posteriorly is the posterior ramus of lateral sulcus. The point where the lateral sulcus divide is called name as the Sylvian point. So let us draw here lateral sulcus. It has three parts: anterior, ascending, and posterior ramus. It has three ramus. This is the superior frontal sulcus. This is the inferior frontal sulcus. This is pre-central sulcus. This is post-central sulcus, and this is the central sulcus. Similarly, there is again two sulcus at the temporal lobe. One is the superior temporal sulcus and another is the inferior temporal sulcus. The superior temporal sulcus extends posteriorly parallel to the posterior rami, and the inferior temporal sulcus also extend posteriorly. Okay. Here is in the parietal lobe there is a sulcus known as intraparietal sulcus, and separating the parietal lobe and the occipital lobe there is a parieto-occipital sulcus, and here is the pre-occipital notch, and in the occipital lobe in the superolateral surface there is a lunate sulcus, and just at the occipital pole there is the post-calcarine sulcus. So here is the superior temporal sulcus, inferior temporal sulcus. Okay, here is the intraparietal sulcus. It is the parieto-occipital sulcus. This is the lunate sulcus, and this is the post-calcarine sulcus. And this is the pre-occipital notch. Now let us discuss the gyrus. So always remember that gyri will be present between two sulcus, and it is also true that one sulci may be present between two gyri. So let us see the gyri. Now we will discuss about the gyri. Remember, the gyri is present between two sulci. So first gyri that we will see is this. It is the pre-central gyrus, and posterior to the central sulcus it is the post-central gyrus. So pre-central gyrus, post-central gyrus. This is the superior temporal gyrus. Between superior frontal sulcus and inferior frontal sulcus there is the middle frontal gyrus, and inferior to the inferior frontal sulcus there is the inferior frontal gyrus. So let us write this. Here is the superior frontal gyrus, here is the middle frontal gyrus, here is the inferior frontal gyrus. Say just like this. Here is the superior temporal gyrus, here is the middle temporal gyrus, and here is the inferior temporal gyrus. Superior temporal gyrus, middle temporal gyrus, and inferior temporal gyrus. This intraparietal sulcus divides the parietal lobe into two gyrus. One is this superior parietal lobule. It is a very big gyrus, that's why it is called as lobule, and the inferior parietal lobule. Superior parietal lobule, inferior parietal lobule. Intervening this there is another gyrus called as angular gyrus. This is the angular gyrus which hooks around the posterior ramus. Now these are the important gyri of the superolateral surface. Now let us see the medial surface. So for the medial surface let us draw a diagram. In the medial surface here is the central sulcus. Above the corpus callosum there is one sulcus that intervenes here, which is known as callosal or the cingulate sulcus, called the cingulate sulcus. Here is the parieto-occipital sulcus. Here is the calcarine sulcus. Here is the collateral sulcus, and here is the occipitotemporal sulcus. Above the corpus callosum there is the callosal sulcus. Callosal sulcus. Then here is the lateral sulcus. Here is the occipito-temporal sulcus. Here is the calcarine sulcus, and here is the parieto-occipital sulcus. Now let us describe the gyri. So here we have the medial frontal gyrus and the medial frontal gyrus. This is the cingulate gyrus. This is the parahippocampal gyrus within the collateral sulcus. Between the collateral sulcus and occipitotemporal sulcus there is the medial occipitotemporal gyrus, and beyond that there is the lateral occipitotemporal gyrus. In front of parieto-occipital sulcus there is the gyrus is known as precuneus. Beyond parieto-occipital sulcus, that region of occipital lobe is called as cuneus, which is divided into two halves by the calcarine sulcus. So let us write the names. Here it is the medial frontal gyrus. This is the cingulate gyrus. This is the precuneus. This is cuneus. This is parahippocampal gyrus. The parahippocampal gyrus is connected to the cingulate gyrus via a small thin segment of white matter which is called as isthmus. This is the medial occipitotemporal gyrus, and this is the lateral occipitotemporal gyrus, and this is corpus callosum. Now let us discuss about the sulci and gyri at the inferior surface of brain. So here I am drawing the cerebral pit. Here I am drawing the midbrain. So this is the inferior surface of the midbrain. So this is the orbital surface, and there is a H-shaped sulci. This is called as orbital sulci. This orbital sulci divides the orbital surface in anterior, middle, lateral and posterior part. So this is the orbital sulci. In the temporal surface, the inferior surface, there are two prominent sulcus. Medial one is called the collateral sulcus. The lateral one is called as occipito-temporal sulcus. Again, there is two more sulci at the occipital pole. One is the linguate sulci or sulcus, another is the post-calcarine sulcus. Now let us discuss about the gyrus. So here we have the parahippocampal gyrus. Lateral to that we have the medio-orbitotemporal gyrus, and more lateral to that we have the lateral orbitotemporal gyrus. Lateral occipitotemporal. I am very sorry, that is occipitotemporal. And the anterior most portion of the parahippocampal gyrus is called as the uncus. So this is the parahippocampal gyrus, medial occipitotemporal gyrus, and lateral occipitotemporal gyrus. So this is all about the gyri and sulci. Now let us move and discuss about the various Brodmann areas or the functional areas of the brain. Brodmann area. Now basically the Brodmann area is basically denotes some of the functional areas of brain. Functional areas of cerebral cortex. It has been seen that these areas differ from each other in structure as well as function. Differ from each other with respect to structure plus function. So total 47 areas are identified and marked by Brodmann. So that's why these areas were known as Brodmann areas. Now, these cortical areas can be broadly divided into three categories. One is the motor area, one is the sensory area, and another is the association area. This association area covers about 75% of total surface of cerebral cortex. Okay, so now let us discuss about various types of areas and their positions in the brain. So first, let us discuss about the motor area. So here is the area number 4. Area number 4, it is the primary motor area, which is present in the precentral gyrus. Then anterior to that, there is the area 6, which is the premotor area. Then which is formed by the posterior portion of superior, middle, and inferior temporal gyrus. Then let us discuss the sensory areas. So 3, 2, 1. This forms the primary somatosensory area. This is present at the postcentral gyrus. So the postcentral gyrus represent the primary somatosensory area. Then the superior parietal lobule represent the Brodmann area 5 and 7. This is the sensory association area, or secondary somatosensory area, or sensory association area. Then coming to the frontal eye field, it is designated as area 8. So the frontal eye field is present at the posterior part of the middle frontal gyrus, and it is anterior to the T-central gyrus. Then coming to the area 44 and 45, these are the motor speech area or the Broca's area. So these are present in the inferior frontal gyrus of dominant hemisphere. Then here is the 41 and 42, here is the primary auditory area, which is present at the superior surface or the superior temporal gyrus, which is also called as Hessel gyrus. Superior temporal gyrus or the Hessel gyrus, which is the primary auditory area. Then the area 22 is the secondary auditory area. Then the sensory speech area, it is called as the 40 and 49, it is called as the sensory speech area. Sometimes this 22 is also called the sensory speech area. Some author mention 40 and 39 as Wernicke's area. Some author mention 22 as Wernicke's area. So be cautious about this. Then the area 19 and 18. The area 17, 18, 19, those are associated with the visual area. Now 17 is the primary visual area and 18 and 19 is the visual association area. Okay, so 17 হচ্ছে primary visual area, 18, 19 secondary অথবা association visual area. Then coming to the medial surface, here is the paracentral lobule. Paracentral lobule is basically this part. Paracentral lobule. It is occupied by this. So this paracentral lobule. So here is the 8, 9, 10, 11. These represent the prefrontal area. And the area number 28 is called the primary olfactory area, which is the anterior part of parahippocampal gyrus. I have discussed about this area in my limbic system video. And last but not the least, that the area number 43 is the taste area, which is present at the inferior part of parietal lobe. Area number 43 is the taste area. So this is in short about the Brodmann area. Discuss about the white fibers of the brain. First, let us know what is meant by a white matter. So the white matter means it is a compact mass of vast number of nerve fibers and associated neuroglia. This term is very important, associated neuroglia. Now, types of white fiber. There are mainly 3 types of white fiber. There is association fiber, commissural fiber, and projection fiber. So let us discuss that on what basis these nerve fibers are classified. So let us suppose this is our cortex or the cerebral hemisphere, and these are the subcortical structures. And these are the cortical structures. So if there is interconnection between the cortical structures of the same hemisphere, then these fibers are known as association fibers. If there is interconnection between one region of one hemisphere with the another region, with the same region or the identical region of the another hemisphere, then this type of connection or this type of white fiber are called as commissural fiber. And if there is connection between subcortical structures, between cortical structures with the subcortical structures, then these type of fibers are called as projection fiber. So now let us see. I have already written this because writing these things, these are many data. You have to memorize this. So it will save some time as it is a one-shot video. So I have already written this. So association fiber are 2 types. One is the short association fiber and another is the long association fiber. So the short association fiber, it interconnects the adjacent gyri. So adjacent gyri interconnection is called as short association fiber. For example, the arcuate fasciculus connecting the Brodmann area 45 with the 39 and 40. So the Brodmann area 45 is the Broca's area and 39-40 is the Wernicke's area. So these arcuate fasciculus is responsible for language generation. Next, the long association fiber. Long association fiber interconnect widely separated gyri. Here are 5 examples. There are 5 types of long association fiber. Uncinate fasciculus, which is this one. Then cingulum, which is the limbic association bundle. I will discussed this association fiber in the limbic system video. So here is this fiber. It starts from the it starts from the hippocampus. It starts from the parahippocampal gyrus. Then it follows the isthmus and then the cingulate gyrus, and then it conveys the information to the septal area. So this is the limbic association bundle. Then the superior longitudinal bundle. Here one of the bundle is the superior longitudinal bundle. Then here is the fronto-occipital bundle, which connects the frontal lobe with the occipital lobe of the same hemisphere, and there is the inferior longitudinal bundle, which connects the temporal pole with the occipital pole of the same hemisphere. Now coming to the commissural fiber, it interconnect identical or corresponding areas of the two hemispheres. So the example here are 5 examples. Corpus callosum, it is the largest commissural fiber of brain. Then there is the anterior commissure, which is formed by the lateral segment of the olfactory trigon. That is forms the anterior commissure. Then the posterior commissure, which will be discussed during the third ventricle video. Then the hippocampal commissure, it is the commissure of fornix. Individual short note of fornix can come in your exam. And then the havenular commissure. So havenular commissure will be also discussed during the third ventricle video. Then we have the projection fiber. So the projection fiber, it connects the cortical areas with the subcortical areas. The subcortical areas include thalamus, hypothalamus, mammillary body, brainstem, spinal cord, etc. So for neocortex, there are two projection fibers are the corona radiata and the internal capsule. And for the allocortex, it is the archae and paleocortex. The projection fibers are the fimbriae and the fornix. So individual short note of fornix may come as a short note. Now coming to the main short note topics are the corpus callosum and the fornix and internal capsule. So from these three, one can come as a short note. These three are the hot topics for your prof exam. Now coming to the corpus callosum, so as I have already told that it is the largest commissural fiber of brain. It connects two cerebral hemisphere and it connects all part of the neocortex except the anterior part of the temporal lobe, which is connected by the anterior commissure. You have to remember that. Now some of the facts, that the length of the corpus callosum is 10 cm. The distance from the frontal pole up to the corpus callosum is 4 cm. So it is present slightly towards the anterior part of the brain, and the distance from the occipital pole to the corpus callosum is 6 cm. Now the superior aspect is covered by a thin sheet of grey matter called as indusium retium. We will discuss with a diagram later. Now here are the respective parts of fornix. First parts of corpus callosum is written here. That is from anterior to posterior. The parts are rostrum, genu, body, and splenium. Okay, here I have written everything which you need to write during your exam. So in order to understand the writing, I will just draw a diagram. So from that diagram you will understand everything. So let us pay attention in the diagram. So I am drawing a 3D view of corpus callosum. So here is the corpus callosum. Now it has some part from anterior to posterior. You repeat with me. That is rostrum, genu, body, and splenium. From the genu, fibers sweeps forward towards the frontal pole into the frontal lobe, and this fiber looks like fork or forceps. So this fiber is termed as forceps minor, because these are small fibers. Again, similarly, there are some fibers which moves posteriorly towards the occipital pole and interconnects the occipital lobe of the two hemispheres. So these fibers are called as forceps. These fibers are called as forceps major. From the body, white fibers interconnect the two hemispheres of the brain. So here is the white fiber. Another special kind of white fiber is there, which is formed by fibers of body and splenium, which is not intersected by the corona radiata. So let us see a drawing. Here is a diagram. Here you can see the red dots represent the fibers of corona radiata, which is oriented in craniocaudal direction, and the fibers of corpus callosum is oriented in horizontal direction. So the fibers which remain unintersected from the fibers from the corona radiata, these forms the tapetum. So these forms the tapetum. So we will now discuss how a tapetum looks. So the fibers that remain uncrossed, it will first passes posteriorly, makes the roof and lateral wall of the posterior horn, then passes forward and downward and makes the lateral wall of the inferior horn. So this is how a tapetum looks. So again I would be telling you that first the fibers of the tapetum passes posteriorly towards the occipital pole, then it makes the roof and lateral wall of the posterior horn of lateral ventricle, then from there it moves downward and forward and makes the lateral wall of the inferior horn of lateral ventricle. So some of the examiner loves to ask the arrangement of tapetum. So this is with a drawing. I try to make you understand how a tapetum looks like. So this is the tapetum. So basically these are the three types of fiber that are extension of the corpus callosum. And one thing that I have forgotten to tell you, that the superior aspect of the corpus callosum remain covered by a thin sheet of grey matter which is termed as indusium racem. Indusium racem. Some clinical points regarding the corpus callosum is that congenital absence or surgical sectioning of the corpus callosum will lead to will behave like that each hemisphere becomes isolated. So it behaves as if the person is having two separate brains. So this is known as split brain syndrome. This is usually done to decrease severe epileptic seizure, and it remains asymptomatic and is only determined after taking an MRI. So this is all about the white fibers of brain. The next lecture will be on the internal capsule. Thank you. Hello everyone, myself Shoradip Nandi, second year MBBS student of IPGM here. So welcome you all to Quizzical Doctors. Today we'll be discussing about the internal capsule. So first of all, internal capsule is a projection fiber which is a subtype of white fibers. So the projection fiber means it will connect the cortical product with the subcortical levels. It has two parts, that is, one is the ascending fiber and it contains the descending fiber. So the ascending fibers are the sensory fibers, whereas the descending fibers are the motor fibers. So these fibers interconnect cortical areas with subcortical areas. That is, the subcortical areas include the brainstem and spinal cord. Now coming to the location of the internal capsule, so it is located between the caudate nucleus thalamus and the lentiform nucleus. So medially there is the head of caudate nucleus and the thalamus, and laterally there is the lentiform nucleus. Now rostrally it fans out to form the corona radiata, and caudal fibers condenses to form the crus cerebri. This is very important to see. So let us see in a diagram. So here is the caudate nucleus and thalamus, and here is the lentiform nucleus. So here I am drawing the fibers of corona radiata, that is the fanning out of the fibers of internal capsule, and caudally it condenses to form the crus cerebri, which continues to the mid brainstem. Now why internal capsule is important to us is that it consists high concentration of sensory and motor fibers. So any small lesion will produce widespread effects, that is sensory and motor loss, which occurs in case of stroke. Now here again I am showing the location. So the internal capsule passes lateral to the head of caudate nucleus, and it passes medial to the tail of the caudate nucleus and reaches the crus cerebri and brainstem. Now coming to the parts and its constituent fiber. So the internal capsule consists of 5 parts, that is anterior limb genu, posterior limb, sublentiform which is SL, and retrolentiform which is RL. Now coming to the parts of internal capsule and its constituent fiber, each characteristic part consists of motor and sensory component of fibers. The motor component of fibers in anterior limb consists of frontopontine, anterior limb genu and posterior limb consist of frontopontine fibers, and the retrolentiform and sublentiform contains the paritopontine and paratopontine fibers. Whereas the retrolentiform consists of occipitopontine because it is present posteriorly, and sublentiform consists of the temporopontine because it is present inferior to the lentiform nucleus level. Again, there are some other fibers. For genu, there are the cortico-spinal tract, and the corticonuclear tract passes through the genu, which supplies the head neck region, and in the posterior limb, the corticospinal tract and the rubrospinal tract passes through the posterior limb. Now coming to the sensory component, there is sensory fiber that passes through the internal capsule. For in the anterior limb there is the anterior thalamic radiation. The genu consists the anterior part of superior thalamic radiation. The posterior limb consists the posterior part of superior thalamic radiation. The retrolentiform part consists the posterior thalamic radiation, also known as the optic radiation. The sublentiform part consists the inferior thalamic radiation or auditory part. There is a diagram which shows the areas from where the pontine fibers are carried. So FP means the frontal region, then from where the pontine fibers are being carried. The pontine fibers are responsible for coordination. And here is another diagram which shows the passage of various thalamic radiation fibers. Thalamic radiation means these are those third order sensory fibers that originate from the thalamus and fans out to go to the various and distribute to the various parts of the brain. In total, it is called as radiation. Arterial supply. Here is the circle of Willis that I am drawing. So here we have the anterior cerebral artery, middle cerebral, internal cavity, anterior choroidal, and the posterior cerebral artery. So from the anterior limb and genu gets its supply from the anterior cerebral artery through the recurrent artery of Hubner. Then the posterior limb gets supply from the middle cerebral artery by the Charcot's artery. The genu also get a supply from the internal carotid artery. The sublentiform and the retrolentiform get supply from posterior cerebral artery. Important to note is that the sublentiform part gets a supply from the anterior choroidal artery. So here is a summary that I am writing, which part gets which artery, which part get which supply from the artery. Now coming to the clinical part, so the clinical part is important for the lesion. So any lesion in the internal capsule will cause widespread sensory and spastic paralysis of contralateral half of the body. So it will result in hemiplegia and hemianesthesia. Now the lesion can be due to two types. One is the hemorrhage and one is the thrombosis of the vessel. Thrombosis lead to ischemia and it leads to neuronal death or brain cell death, which is irreversible. So one example is the anterior choroidal artery. It is also called as artery of cerebral thrombosis. So its lesion will lead to the lesion of sublentiform part of the internal capsule, which will lead to auditory problems and loss of coordination. And in case of hemorrhage, the Charcot's artery, it is the branch of middle cerebral artery. It is also called as artery of hemorrhage. It supplies the posterior limb. So if there is some hemorrhage and it will cause damage to the posterior limb, it will cause contralateral hemiplegia and hemianesthesia. So this is it for internal capsule. Remember, internal capsule is a very important topic for your university exam, and it comes regularly in the exam. So prepare accordingly. You will be. So now let us discuss about the ventricular system. So ventricles are the cavities in the CNS through which cerebrospinal fluid flows. There are 4 ventricles inside the brain: 2 lateral ventricle, third and fourth ventricle. If we are told that what are the number of ventricles in the CNS, so there are 5, which so additional is the terminal ventricle. Now I am drawing here the overall overview of the ventricle. So here is the lateral ventricle, third and the fourth ventricle, and at the terminal portion of the spinal cord there is the terminal ventricle. Okay. So this is the overview of the ventricle, how it is present inside the CNS. Now we will start with our lateral ventricle. So lateral ventricle, it is the cavity present within the cerebral hemisphere. Now the site, it wraps around the thalamus, lentiform nucleus and the caudate nucleus. It is separated. The 2 lateral ventricles are separated by septum pellucidum, which is a thin vertical sheet of white and grey matter. Now, now the 2 lateral ventricle communicate with each other by interventricular foramen of Monro, communicate with each other, and the third ventricle by interventricular foramen of Monro. Now, coming to the parts of the lateral ventricle. So there are 4 parts of lateral ventricle, that is anterior horn, body, posterior horn and inferior horn. Now the demarcation, the body extend from the interventricular foramen of Monro up to the splenium of corpus callosum. Sorry, it is the body, interventricular foramen of Monro up to the splenium of corpus callosum. Now what is anterior horn? Anterior horn is the anterior extension of the body of lateral ventricle up to the posterior surface of genu of corpus callosum. Then the posterior horn, it is the posterior extension of the body of corpus callosum towards the occipital pole. And the inferior one, it is the largest among all the parts. It is the direct continuation curve. It is the direct continuation of lateral ventricle. It curves round the pulvinar of the thalamus and enters the temporal lobe and moves towards the pole. So that is the inferior horn. Now coming to the boundaries. So each section of each part has a definite coronal section. So in case of anterior horn, there is roof, floor, anterior wall, medial wall and the lateral wall. So here I have drawn the parts. So as you can see from the diagram, the roof is formed by the under surface of anterior part of the body of corpus callosum. The floor is formed by the upper surface of rostrum of corpus callosum. The anterior wall is formed by the genu of corpus callosum. Medial wall is formed by the septum pellucidum, and the lateral wall is formed by the head of the caudate nucleus. So the diagram is like a coronal section, taking a coronal section from this part. Now let us see what are the boundaries of the body. The boundaries of the body of lateral ventricle come as a short note in your prof exam, so prepare it accordingly. So here is the corpus callosum. Here within the septum pellucidum we have the fornix. Here I am drawing the tela choroidea of the lateral and third ventricle. Tela choroidea is the part through which the CSF is produced. Now here I am drawing the thalamostriate vein. So here is the choroid plexus, here is the thalamostriate vein, then here is the stria terminalis, then here is the body of caudate nucleus. So the roof is formed by the under surface of body of corpus callosum. The medial wall is formed by the septum pellucidum. In the floor is formed from lateral to median, the body of caudate nucleus, stria terminalis, thalamostriate vein, choroid plexus and the upper surface of body of fornix. Now coming to the posterior horn, the boundaries of the posterior horn. So here I am drawing the diagram. Okay, so I have drawn the diagram. So the roof, lateral wall and floor is formed from inside outward by the tapetum, then optic radiation and the inferior longitudinal fasciculus. In the medial wall is formed by the forceps major and an indentation formed by the calcarine sulcus on the medial wall of the posterior horn, which is called as calcar avis. It is an indentation formed by the calcarine sulcus. Now coming to the inferior horn, here I am drawing the diagram. So the inferior horn is present in the temporal lobe. So here I am drawing the choroid plexus and the inferior horn of lateral ventricle. All these are coronal sections. Okay. So here is the tapetum, here is the auditory radiation, and here is the inferior longitudinal fasciculus. So, and here is the collateral sulcus, which forms an indentation on the floor of the inferior horn called as collateral eminence. Here is the auditory radiation, here is the tapetum. It will be the auditory radiation here. And here is the tail of caudate nucleus, here is the stria terminalis. This is the choroid plexus of the inferior horn. This is the fimbria of fornix. This is the hippocampus. So now let us write the boundaries here. So the roof from medial to lateral is formed by stria terminalis, tail of caudate nucleus and tapetum. Also the lateral wall is formed by the tapetum, roof and the lateral wall. Now the floor is formed by the, from medial to lateral, it is formed by the hippocampus and collateral eminence. So now let us discuss about the third ventricle. So the third ventricle is a cavity present in between thalamus and hypothalamus. That is, it is a cavity present in the diencephalon of both the hemisphere. So here I am drawing the diagram of the third ventricle. So here is the corpus callosum. Now I am drawing the optic chiasma. The below the corpus callosum, it is the fornix. Behind the fornix, there is the pineal gland with its stalk, and here I am drawing the brainstem and cerebellum. So here I am drawing the choroid plexus of the third ventricle. Here is the thalamus, here is the hypothalamus, and below the hypothalamus there is pituitary and the mammillary body. Now the upper stalk of the pineal gland consists the habenular commissure. So it has the upper stalk and the lower stalk. Through the upper stalk, the habenular commissure passes, which connects the habenular nucleus of the thalamus of both sides, and through the lower stalk, the posterior commissure passes, which connects the two superior colliculus of both sides. So now I am drawing the demarcation of the third ventricle with dotted lines. So here you can very well see how the third ventricle is present inside the CNS and how it communicates with other ventricles. So now I will discuss about the boundary. So here is the optic chiasma. In front there is the lamina terminalis through which passes the anterior commissure. Here is the pituitary gland. Then posteriorly there is the mammillary body. And between optic chiasma and pituitary there is an elevation in the floor of the third ventricle, which is known as tuber cynarium. So now let us write the boundaries. So the roof is formed from foramen of Monro, interventricular foramen of Monro, through which it communicates with the lateral ventricle. So the extent of roof, it is from the foramen of Monro up to the habenular commissure. So the roof is formed by two things. One is the fornix, that is the under surface of the fornix, and the telachoroidea of third ventricle. Telachoroidea of the third ventricle. Now the posterior wall, it is formed by the upper stalk of the pineal gland containing the habenular commissure, the pineal gland itself, and the lower stalk of the pineal gland containing the posterior commissure. So the diagram is like this. That is the posterior wall. Now coming to the anterior wall, it is formed by the lamina terminalis containing the anterior commissure and the anterior column of fornix. Anterior column of fornix. So coming to the floor, it is formed from anterior to posterior by optic chiasma, tuber cynarium, infundibulum of the pituitary, mammillary body, and the posterior perforated substance. So here lies the posterior perforated substance. This posterior and anterior perforated substance will be discussed during our cerebral circulation video. The lateral wall, it is formed by the thalamus, hypothalamus, and interthalamic adhesion. What is interthalamic adhesion? So these are the fibers of the thalamus that tries to go to the opposite side but revert back to its original position. So these are the interthalamic adhesions. So now let us discuss about the recess of the third ventricle. So number one recess is the vulva. Second one is present superior to the optic chiasma, so it is called a supraoptic. Then third one, which is present just at the infundibulum of the pituitary, so it is infundibular. Fourth one is present above the pineal gland, so it is suprapineal. And final, the fifth one is present just anterior to the pineal gland, so it is called as pineal recess. So 5 recesses are there. I have drawn a diagram. So now let us discuss about the fourth ventricle. So fourth ventricle is a tent-like cavity in the hind end, that is the rhombencephalon. So the location, it is present in the posterior cranial fossa, in front of the cerebellum, pons, and upper part of the medulla. Now the shape, in sagittal section it looks like a triangular cavity, and in coronal section it looks like a rhomboid cavity. So here I am drawing the diagram. So here is the fourth ventricle, which in sagittal section looks like a triangular cavity, and in coronal section it looks like a rhomboid shape. Now coming to the boundaries. So here are the two inferior colliculus from where the superior cerebellar peduncle are forming. So the superolaterally it is bounded by the superior cerebellar peduncle, and inferolaterally by the inferior cerebellar peduncle. Now forming the roof or the posterior wall, it is formed by upper sloping surface, apex, and lower sloping surface. So the upper sloping surface is formed by the convergence of the superior cerebellar peduncle, which is also known as the superior medullary velum, which is a white nervous tissue. So superior medullary velum is the white nervous tissue. On it lies the lingula part of the vermis. The apex, it extends posteriorly into the white matter of cerebellum, and the lower sloping surface is formed by the inferior by the pia mater and the ventricular ependyma. Together it is called as inferior medullary velum. There are 3 openings there, two laterally, which are called as foramen of Luschka, and one posteriorly on the roof, that is the foramen of Magendie. Now coming to the diagram for the floor of the fourth ventricle. So here I am drawing the diagram. So here is the superior cerebellar peduncle, middle cerebellar peduncle, and the inferior cerebellar peduncle. So here it is the superior colliculus, inferior colliculus of midbrain. There is the median sulcus. Here is the locus coeruleus. Here is the superior fovea. Here is the facial colliculus formed by the movement of the facial nerve. Here is the vestibular area. Here is the sulcus limitans. Here is the stria medullaris. Here is the tinea. And here is the inferior fovea. Here is the vagal triangle. Here is the area postrema, which is responsible for the vomiting center. Here is the middle cerebellar peduncle, inferior cerebellar peduncle, and the superior cerebellar peduncle. Here is the 2 tubercle, that is the cuneate and the gracile tubercle. Medially it is the gracile, laterally there is the cuneate tubercle. So this is all about the floor of the lateral ventricle. It might come as a short note in your prof exam. Clinical. So clinically there is, due to increased cerebrospinal fluid pressure in the brain, either due to increased production or obstruction, the condition is known as hydrocephalus, and that. So now we will discuss about the limbic system. So the limbic system is associated with olfaction and its effect, emotion, and memory. One dictum you have to remember, that the sensation must reach the hypothalamus. So here I am drawing the components of the limbic system. So here is the corpus callosum. Here I am drawing the caudate nucleus. The tail of the caudate nucleus contains the amygdaloid body. Here is the thalamus. Below the thalamus there is the hypothalamus. The part of hypothalamus there is the mammillary body. Now here I am drawing the hippocampus. I will be drawing the hippocampus, which looks like the paw of a tiger. Here it is the cingulate sulcus. Below the cingulate sulcus there is the cingulate gyrus, which is continuous with the parahippocampal gyrus. So the cingulate gyrus is continuous with the parahippocampal gyrus. So here the diagram is complete. So first, the sensation of olfaction is carried by the olfactory nerve, and then the sensation is taken by the olfactory bulb present over the ethmoidal plate. The olfactory bulb then forms the olfactory tract, and the olfactory tract, approaching towards the anterior perforated substance, divides into 3 divisions and forms the olfactory trigone. These 3 divisions are named as lateral, middle, and medial olfactory stria. So here is the ethmoidal plate through which the optic nerve carries the information. Now the medial stria joins to form the anterior commissure. The medial olfactory stria joins to form the anterior commissure. The lateral olfactory stria, the fiber goes to the primary olfactory area, that is Brodmann area 20. From the primary olfactory cortex, fiber goes to amygdala and parahippocampal gyrus. goes to parahippocampal gyrus and amygdala. From amygdala, I have represented the amygdala with A. From amygdala, striate terminally starts. It winds round lateral to the caudate nucleus, approaches the anterior commissure, divides into two divisions. One goes to septal area, which is the precommissural fiber. Another goes to the hypothalamus. So our dictum is clear that the sensation must go to hypothalamus, so it has reached the hypothalamus. Now from the parahippocampal gyrus, which is the second olfactory area, fiber goes to hippocampus. There starts the fiber of fornix, that is the fimbri, making a sheet over the white matter over hippocampus called as alveus. Then the fibers of fimbria condenses to form the crus of fornix. Then the body of fornix encircles over the thalamus, approaches the anterior commissure. On reaching the anterior commissure, it divides in two fibers, that is precommissural and postcommissural. Precommissural fiber relays over the septal area, and the postcommissural fiber goes to three regions. One is the hypothalamus, second is the mammillary body, and third is the tegmentum of the midbrain. So here is the pathway through which the sensation of olfaction reaches the hypothalamus. So here I am writing all the labelings. Lateral stria is the primary olfactory cortex. Now here I have drawn the flow chart which I have spoken through my mouth, that the olfactory tract approaching towards the anterior perforated substance divide into three divisions: lateral, medial and medial. The lateral stria fiber goes to a primary olfactory cortex, and the medial stria joins to form the anterior commissure. So bulb, tract, trigone and stria. From primary olfactory cortex, fiber goes to amygdala and parahippocampal gyrus. Now there are two pathways. So from amygdala, stria terminally start. It winds round the caudate nucleus, reaches the anterior commissure, divides into division pre and postcommissural. Precommissural goes to septal area and postcommissural goes to hypothalamus. From parahippocampal gyrus, fiber goes to hippocampus. Then there starts the fimbria, making a thin sheet of white fiber over the hippocampus called alveus. Fiber of fimbria then condenses to form the crus of fornix, then the body of fornix. Then on reaching the anterior commissure, divided into precommissural postcommissural fiber, which goes to the designated area. So here is the summary. Then from the body of fornix, it makes the column. Now here is the Mbbs circuit. So Mbbs circuit is a circuit responsible for memory generation. So once some smell is sensed by the hypothalamus, that is the mammillary body, that sensation is sent to the thalamus by the mammillothalamic tract. Then from thalamus, it is sent to different areas. From anterior nucleus of the thalamus, it goes to cingulate gyrus. So I am drawing here the pathway. So this is the mammillary body. From here, the sensation goes to anterior nucleus of thalamus by mammillothalamic tract. From the anterior nucleus of thalamus, it goes to cingulate gyrus. From cingulate gyrus, the sensation goes to parahippocampal gyrus by a specialized association fiber which is named as cingulum, by a long association fiber called as cingulum. So from cingulate gyrus, sensation goes to parahippocampal gyrus. The cingulate gyrus is connected to parahippocampal gyrus by the isthmus, and the sensation is carried to the parahippocampal gyrus by cingulum, which is a long association fiber. So this whole of the cycle is repeated multiple times, and this is responsible for the memory generation. So that if that smell again, then it looks familiar. So here I am drawing the summary. Then from parahippocampal gyrus to hippocampus, then through fornix, it again comes back to mammillary body, and this cycle is repeated. Okay, now discuss about the fornix. Now fornix is a white fiber of brain. It is mainly a projection fiber, but it consists of all the three types of fiber, that is association, commissure as well as projection fiber. It starts its journey from hippocampus and ends its journey in septal area, hypothalamus and tegmentum of midbrain. That is, hypothalamus and tegmentum of midbrain are the subcortical areas, whereas hippocampus is a cortical area. So it interconnects cortical with the subcortical area, so it is a projection fiber. Remember, I told in the white matter video that fornix is the projection fiber of allocortex. Now coming to the other fibers of fornix, that is the association fiber. So why it is the association fiber? Because it connects the hippocampus of one hemisphere to the septal area of the same hemisphere. So it connects one region of brain to the another region of the same hemisphere. That's why it is a association fiber. Second, As I have discussed earlier, it is a projection fiber because it connects hippocampus, which is the cortical area, to the hypothalamus and tegmentum, which is the subcortical area. So it is a projection fiber. Now, coming to the commissural fiber of fornix. So the commissural fiber is that often fibers between two fornix of two hemisphere shares information between the hippocampus of the two hemisphere. Often the fibers between the two fornix of the two cerebral hemisphere shares information between the hippocampus of the both hemispheres. So it behaves as a commissural fiber also. So here is a diagram that you can draw during your prof exam. So here is the fornix and here is the hippocampal commissure. So hippocampal commissure is a part of the fornix. So fornix is an important short note for your university exam. So prepare it with utmost importance. Fornix, internal capsule and corpus callosum, three are the most important with the spinal cord. So the spinal cord is the lower extension. Spinal cord is the lower elongated part. Of CNS. Now it is slightly flat anterior-posteriorly and occupies the upper 2/3 of the vertebral canal. So let us now see where a spinal cord starts and where it ends. So this is the brain. Here is the brainstem structures. Here is our cerebellum and the spinal cord starts from here. So in adult the spinal cord starts from an imaginary plane that passes from three structures. So the spinal cord starts from an imaginary plane that passes from three structures. Number one, just rostral to the attachment of first pair of spinal nerve. Just rostral means just cranial to the attachment of first pair of spinal nerve. Second, upper border of posterior arch of atlas vertebra. And third is the middle of odontoid process of axis. So these are the three landmarks from where the spinal cord starts. That is just rostral to the attachment of the first pair of spinal nerve, upper border of posterior arch of atlas, and middle of the odontoid process. Where the spinal cords end. So the spinal cord ends as a tapering end called as conus medullaris at the level of intervertebral disc in between L1 and L2 vertebra. In anatomy you have to be very clear in your language. So spinal cord ends as a tapering end called as conus medullaris. So this tapering end is called as conus medullaris, and where the conus medullaris is present at the level of intervertebral disc in between L1 and L2 vertebra. Most of you will utter the word that the spinal cord ends at the lower border of L1 vertebra. That is somewhat like a layman's term, but anatomically you must utter that it ends as a tapering end called as conus medullaris at the level of intervertebral disc in between L1 and L2 vertebra. Okay, so this is a gross morphology of spinal cord. Now let us discuss the positional changes of spinal cord. Positional changes of spinal cord. Positional changes means at third month of intrauterine life, the spinal cord extended up to the first coccyx vertebra. Then gradually vertebral growth was faster than that of spinal cord growth. So because of this, the vertebral length increase, but the spinal cord length does not increase, and the lower level of the spinal cord gradually starts increasing. Towards the lumbar vertebra. So at birth, it remains at the H2 vertebra, lower border of H2 vertebra. Then in adult, it comes to the L1 vertebra, lower border of L1 vertebra. So this knowledge of variation of positional change of spinal cord is important important with respect to lumbar puncture. You have to determine that at what level the lower extent of the spinal cord is, else you may end up damaging the spinal cord and it would be detrimental for the patient. So lumbar puncture is usually done below L2 vertebra because up to L1 spinal cord lower limit. This is for adult. So in children, usually the spinal cord is extend more downward than that of L1. It might be L3, it might be L4. Now we will go to our discussion with the spinal modification or covering or the modification of the pia mater. So now we will discuss about the covering and meninges. So you know that meninges have three layers from outside inward. It is the dura mater, then arachnoid matter and the pia matter. In case of spinal cord, the dura matter is the meningeal layer of the dura matter. The arachnoid matter continues from the cerebrum or the brain itself, but there are some modification of pia matter in case of spinal cord. Among those modifications, I will be discussing, there are about 4 modifications. So the spinal pia is the continuation of the meningeal layer or the cerebral pia. Laterally, it gives one pair of modification which looks like tooth-like projection. So this modification is called as ligamentum denticulatum. So because it looks like a teeth-like appearance. So there is one sheet on each side and each sheet contains 21 tooth-like projection. Now I am drawing here the vertebral space, the vertebral canal on inside which there is a spinal cord. Here I am drawing the spinal pia. So laterally it gives the ligamentum denticulatum. In anteriorly, both sides of pia cross each other and forms linea splendis. This crisscross arrangement of pia matter is called as linea splendens. Now posteriorly, there is a septum that attaches the pia matter with the subarachnoid or the arachnoid matter. This modification of pia matter is called as subarachnoid septum because it joins the pia with the arachnoid matter. Fourth modification is the phylum terminal. How phylum terminal is formed, I will discuss later. So the 4 modifications of pia matter are the ligamentum denticulatum, linea splendens, subarachnoid septum, and phylum terminal. Importance of ligamentum denticulatum, it is a guide for the neurosurgeon because it helps to know the position of sensory and motor root. Anterior to the ligamentum denticulatum, the root is the motor root, and posterior to it is the sensory root. Coming to phylum terminal. So phylum terminal is formed because the spinal cord in the intrauterine life extend up to the first coccyx, but gradually when the vertebral growth exceeds the spinal growth and the vertebral length becomes more and more longer than that of the spinal cord gradually ascend up, but the phylum terminal remain at the level of first coccyx. Again, the dura matter remains also at the level of first coccyx, but the arachnoid matter ascends up to the second sacral vertebra, and the spinal cord in adult finally remain up to the L1 vertebra. So here I am writing all the information that the spinal arachnoid ends up to the level of S2, lower border of S2, and the spinal dura ends up to the level of lower border of first coccyx. So therefore, if we take section above S2, we can find all the 3 layers of the meninges. But if we take section in below S2, we can see only 2 layers, that is the dura and the pia matter. So the part of phylum terminal above S2 is the phylum terminal internum, and below S2 it is the phylum terminal externum. Now let us see the how are the spinal roots are formed. So here is the posterior root that is entering through the posterior horn. Here is the anterior root which is exiting through the anterior horn. Both the root takes the dura and the arachnoid matter with itself, and before leaving the and the dura and arachnoid matter follow the nerve root up to the intervertebral foramen till they join to form the spinal nerve. So this is up to which the meninges is present, up to the intervertebral foramen. So you can tell that CSF will flow up to the intervertebral foramen because the CSF flows within the subarachnoid space. Now let us discuss how a spinal nerve is formed. So the spinal nerve is formed by taking the motor root and the sensory root. The motor and the sensory root joins together to form the spinal nerve, and the spinal nerve, after leaving the intervertebral foramen, divides into 2 rami. One is the larger ventral rami and the smaller dorsal rami. Both the rami contains both component of the spinal nerve, that is sensory and motor component. That is why spinal nerve is called the mixed nerve, because be the rami or the spinal nerve both contains both of the component, that is sensory and the motor component. Now let us discuss about the blood supply of spinal cord. So the spinal cord receives its blood supply from the vertebral artery which arise from the first part of the subclavian artery. So here I am drawing the brainstem structure, and here is the spinal cord, here is the foramen magnum. Now here is the vertebral artery, its first part, second part, the first part, second part, here is the third part, finally the fourth part or the intercranial part of the vertebral artery. Both the vertebral artery of both sides joins to form the basilar artery in front of pons. See, here I am drawing the parts of the vertebral artery. Now from the fourth part of vertebral artery, the spinal artery is formed. From the anterior portion, from the two sides of vertebral artery, that is right and left vertebral artery, branches come and joins anteriorly to form the anterior spinal artery. And from left vertebral artery, there originate the left posterior spinal artery, and from right vertebral artery originate the right posterior spinal artery, which descend down and supply the spinal cord. Now, considering a cross section of the spinal cord, we can see anteriorly at the anterior fissure, we can see the anterior spinal artery, and in the posteriorly at the posterolateral sulcus, we can see the posterior spinal artery. The anterior spinal artery supplies the anterior 2/3 of the spinal cord, whereas the posterior spinal artery supplies the posterior 1/3 of the spinal cord. Now, the vertebral artery is not long or bold enough to supply the entire spinal cord. So, in order to supply the entire spinal cord, there needs some assistance from other arteries. So, it needs some support branches. In the cervical part, the cervical arteries give the support. In the thoracic part, the posterior intercostal artery gives the support, and in the lumbar part, the lumbar arteries give the support. As we move caudally, the arterial contribution from vertebral arteries gradually decreases. So, in lumbar region, the supporting branches of lumbar arteries are bold and strong enough to supply that region. That is why these arteries are called as arteria radicularis magna. In the lumbar region, the supporting branch of lumbar artery are strong and bold enough to supply that region, so those arteries are called as arteria radicularis magna or artery of Adamkus. This might be asked in your MCQ or in case of Viva. So let us see this with a diagram. In the anterior view, we can see gradually the contribution from the vertebral artery gradually diminishes, and in the posterior view also. So here are the supporting arteries. Here the cervical. In the cervical region, in the thoracic region, there is the posterior intercostal artery, which support by doing gradual anastomosis, and in the lumbar region, there is the lumbar artery. So caudally, there is maximum contribution from the lumbar arteries, and cranially, there is maximum contribution from the vertebral artery. So, clinical part, that anterior spinal artery, it supplies the anterior 2/3 of the spinal cord. If there is any blockage, it can lead to compromisation of anterior 2/3 of the spinal cord and can lead to motor disturbances. Now, coming to the hemisection of spinal cord, which is also known as Brown-Sequard syndrome, I have drawn the spinal cord at two levels. Let us see what are the normal pathway or the normal tracks. First, see the corticospinal tract. Here is the upper motor neuron comes here, then it gives supplies on the lower motor, relays on the lower motor neuron, and the lower motor neuron comes out of the spinal cord. Now, seeing the spinothalamic tract, first the first order neuron comes, relays over the second order neuron, and the second order neuron ascends up through the spinothalamic tract. Coming to the posterior column, the medial lemniscus pathway, so it directly enters the spinal cord and directly ascends up. Blue is the dorsal column, red is the corticospinal tract, and green is the spinothalamic tract. Now let us see what will occur if half of the spinal cord is damaged. Let us see this level is damaged. So lesion at the level and below the level. So prepare a chart like this, that what will be effect on ipsilateral side, contralateral side, and at the level and below the level. So you can see at the level of corticospinal tract there will be ipsilateral lower motor neuron palsy, and the contralateral side is all good, and below the level there will be upper motor neuron palsy and the contralateral side is all good. Spinothalamic tract, at the level of lesion, there the ipsilaterally it will be lost as well as contralaterally, because the crossing will not take place, but below the level it is all good, but the contralateral fiber which has already crossed cannot reach or ascend up because of the lesion. So the contralateral spinothalamic tract is lost. There will be contralateral hemianesthesia. and ipsilateral hemianesthesia at the level of lesion. Coming to the dorsal column medial lemniscus pathway, there will be at the level there will be ipsilateral lesion. Also below the level there will be ipsilateral lesion, but the contralateral dorsal column tract will escape the lesion. I hope you can understand from this chart. Let us see what are the clinical features. So at the level, it usually occurs at the hemisection of spinal cord usually occur at the thoracic and lumbar level. So I am drawing like this. So at the level there will be loss of spinothalamic tract. So pain, temperature, pressure and cruise touch, all the sensation will be lost here. So and there will be contralateral hemianesthesia, and there will be ipsilateral upper motor neuron palsy below the lesion, and at the level of lesion there will be motor loss, which is the lower motor neuron type. Contralaterally there will be hemianesthesia, and there will be ipsilateral loss of fine touch and proprioception and joint and vibration sense, and there will be contralateral hemianesthesia. So in this chart basis approach and that diagram, it will help you to understand and remember the Brown-Sequard syndrome or the hemisection of spinal cord. You need not to memorize everything, just remember the pathway. So what are the clinical features we will be getting? Contralateral hemianesthesia, ipsilateral motor loss, lower motor neuron and upper motor neuron type, and ipsilateral fine touch proprioception is lost. So this is all about the spinal cord. Discuss about the brainstem section and its lesion. So brainstem includes midbrain, pons and medulla. So here we have a picture of the brainstem. So our topic is brainstem section, that how the brainstem looks at different sections and its lesion. So the brainstem include the midbrain, pons and medulla. So we will be taking sections from spinal cord up to the midbrain. So the section of these, the first one is the spinal cord. So here we have the section. Then the lower closed medulla. Closed means that that it is not exposed to any ventricles, only the central canal is present. So here is the lower closed medulla, here is the upper closed medulla. Then here is the open medulla. Open medulla means that it is exposed to ventricle. That is, here is the fourth ventricle. Then the lower, now coming to the pons. So in medulla we have three sections. In pons we have two sections. One is the lower open pons. So here it is open behind there is the fourth ventricle, and upper closed pons. So here is the closed part of fourth ventricle. Then coming to the midbrain, we have there are two sections. One is the midbrain at the inferior colliculus level and at the superior colliculus level. So now let us draw the structures. So first let us draw the spinal cord. So here is the central grey matter of spinal cord. Here is the central canal. Let us draw the central canal here also. And then here also we have the central canal. Then here we have the fourth ventricle behind. So here is the fourth ventricle. Here is also the fourth ventricle. Here is the fourth ventricle in closed manner. Then here we have the cerebral aqueduct. Here also we have the cerebral aqueduct. Now now in the midbrain, you already know there is a structure called as substantia nigra. Here is a structure. It is named as substantia nigra. It is present in the midbrain at both superior and inferior colliculus level. Now first we have the corticospinal tract. So the corticospinal tract coming first starting from the pyramidal layer of the cerebral cortex, then it passes through the posterior limb of the internal capsule, and then it comes into the cerebral peduncle. So then it passes to the crus cerebri. So this part is the crus cerebri. Okay, so here it passes here. So here are the fibers of the corticospinal tract, both right and left fibers. So here we will also see the corticospinal tract. Coming to the pons, this fiber dissociates. It spreads out like this. Same in this. Now in the open medulla, we will find here is the corticospinal tract. Let us draw a bit like this. Now at the lower now at the upper closed medulla, here is the corticospinal tract. In the lower closed medulla, the tract interchange, that is, it decussates. So first here is our grey matter. Here is the grey matter, and the fibers of corticospinal tract decussates. That is, it cross to the opposite side. It crosses to the opposite side. So it causes a division. It causes a division of the grey matter here. So very small part comes down as the anterior corticospinal tract, and maximum part of it comes down as the lateral corticospinal tract. Okay, so we have been completed with the corticospinal vein. Now comes to the corticopontine. So corticopontine. So corticopontine fiber, again it originates from the pyramidal layers of the cerebral cortex. Then it passes through the internal capsule and it comes at the crus cerebri. So here is the crus cerebri. So again we will draw it. So corticopontine fibers are abundant fibers. It is present in more amount than that of corticospinal tract fibers. So it descends down like this. Coming to the pons, it again dissociates. In the lower open pons, it relays on the pontine nucleus. And these nucleus supply the contralateral cerebellar hemisphere through middle cerebellar peduncle. So these are the middle cerebellar peduncle, these are the superior cerebellar peduncle, and this is the inferior cerebellar peduncle. So the pontine fibers forms the middle cerebellar peduncle. Okay, now let us discuss the dorsal column and medial lemniscus pathway. So the dorsal column, so here is the funiculus gracilis and here is the funiculus cuneus. Gracilis and cuneus. So here is the gracilis and here is the cuneus. So it ascends up. At the level of upper closed medulla, it relays on gracilis and cuneus nucleus present in the upper closed medulla. So these are the gracilis and the cuneus nucleus. From the gracilis and cuneus nucleus, it forms the medial lemniscus tract. So gracilis comes anteriorly. The cuneus comes. The gracilis comes anteriorly. And the cuneus they are posteriorly. Now this gracilis, this is the medial lemniscus tract, again ascends up, comes into the open medulla, then it comes into the lower open pons, then it comes into the upper closed pons, then it occupies the medial portion. Then it occupies the medial portion of the midbrain at the inferior colliculus level, and then at the midbrain at the superior colliculus level. So there is also the red nucleus present anterior to the medial lemniscus. So the dorsal column medial lemniscus tract is now completed. Now we will discuss about the spinocerebellar tracts. So there are two spinocerebellar tract, that is anterior and posterior spinocerebellar tract. The posterior spinocerebellar tract receive information from the ipsilateral side, and anterior spinocerebellar tract receive information from the contralateral side. So both receive the information of proprioception, joint and vibration sense. So it ascends up through the lower closed medulla, then the upper closed medulla, then at the level of open medulla, the posterior spinocerebellar tract approaches towards the cerebellum and enters into the cerebellum via internal cerebellar peduncle. So the inferior cerebellar peduncle is formed by the posterior spinocerebellar tract. So here is the anterior spinocerebellar tract. It ascends upward, and in the superior cerebellar peduncle, and at the upper closed pons level, it crosses the direction and supply the contralateral side of cerebral hemisphere. So let us again discuss the spinocerebellar tract. I think that it is going to be to be a bit confusing to you. That for you need to understand the posterior spinocerebellar tract. It carries the information from the ipsilateral side. So this tract carries ipsilateral information, and coming to the open medulla, it ipsilaterally goes to the designated cerebellar hemisphere. So you all know a term that cerebellum is always ipsilateral. Its input is also ipsilateral. Its output is also ipsilateral. So here the posterior spinocerebellar tract, as it is taking information from the ipsilateral side, it does not need to be crossed. So it goes to the designated cerebellar hemisphere, forming the inferior cerebellar peduncle, whereas the anterior one moves more upward, and at the level of upper closed pons it crosses the midline, that is, it decussates and goes to its designated cerebellar hemisphere. Because you know that the anterior spinocerebellar tract receive information from the contralateral side of the body, so two times taking information from the opposite side of the body, then again crossing and going to the opposite cerebellar hemisphere. So two times crossing makes the information ipsilateral. I hope you can understand the concept. So now let us come to the spinothalamic tract. So here we have two spinothalamic tract. One is the lateral spinothalamic tract, another is the anterior spinothalamic tract. So both ascends up, and both the lateral and anterior spinothalamic tract approaches towards each other, and it becomes very close to each other, and it becomes merged with each other. So here is the spinothalamic tract, the lateral and the anterior spinothalamic tract together merged to form it will form in the midbrain the lateral lemniscus. So then again it continues its journey through the pons, again as the lower open pons and the upper closed pons. Then here it occupies somewhat lateral position, and at the level of superior colliculus it is also named as the spinal lemniscus or lateral lemniscus. At the midbrain it forms a spinal lemniscus. So our major tracts has been discussed. Now let us discuss the various entries and various levels of the cranial nerves. So first at the level of open medulla, three nerves exits. That is 9, 10, 11. Cranial nerve 9, 10, 11 exits through different levels because it has a common nucleus called as nucleus ambiguous. Here we have the olivary nucleus. This is the inferior olive. Here is a presence of a nucleus which is called as arcuate nucleus. It has two types of fiber. The one type of fiber passes to the external part of the open medulla and enters the cerebellum. These fibers are called as external arcuate fibers. And another type of fiber, it goes through the substance of the medulla, then goes through the posterior surface of the medulla to supply the cerebellum. This is called the internal arcuate fibers. These are the internal arcuate fibers. Now, from the lower open pons, there are two cranial nerves that exits. Two cranial nerves that exits. One is the sixth cranial nerve, which is the abducens nerve. And another is the seventh nerve, that is the facial nerve. So it winds round around the abducens nucleus and exits from the pons. So it makes these bulges which are known as facial colliculus. Now coming to the third and fourth nerve. So the third nerve, superior colliculus. So the third nerve originates from the third nerve, that is the oculomotor nerve. So it has the oculomotor nucleus here. So it exits ipsilaterally by passing through the red nucleus, the substantia nigra, and the corticopontine fibers, and finally outside. This is the cranial nerve 3. Cranial nerve 4, which is the trochlear nerve. It crosses before exiting. So this is the only nerve which crosses before coming in, emerging out of the brainstem. Now what about the fifth nerve? So remember the trigeminal nerve. Enters the brainstem into the upper closed pons. So it enters the brain at the level of upper closed pons. The trigeminal nucleus, which crosses the midline. So here it makes the trigeminal lemniscus. Some fibers of trigeminal passes inferiorly and forms the spinal nucleus of trigeminal. At least up to the upper closed midline. the brainstem lesion. So the brainstem lesion, so it is important for the declaration of death. Because the brainstem death is declared as the brain death. So how you can declare that it is a brainstem death? That is by the absence of corneal reflex. So corneal reflex, you have to know the pathway. That in corneal reflex, the afferent signal from cornea is taken towards the brainstem by fifth cranial nerve, that is the trigeminal nerve, and there is blinking of the upper eyelid. So that signal is sent through by the facial nerve, that is the cranial nerve 7. So that is the pathway of corneal reflex. Another lesion that can occur is the brainstem stroke, which is due to either occlusion, that is thrombosis of the arteries, and it can also occur due to hemorrhage of the artery as well. So let us see what part it may cause lesion. So here I am drawing the open medulla. So here is the open medulla. One thing I am also drawing, here is I am drawing the brainstem. Here is our cerebellum. So I am dividing the cerebellum in superior and inferior portion, and I am dividing the inferior portion of cerebellum into anterior and posterior portion. So now we have the here is the foramen magnum. We have the two vertebral arteries that enters towards the brain through the foramen magnum. It then passes anteriorly and joins with the vertebral artery of the other side to form the basilar artery. So before joining, when it is in the posterior portion, it gives branches to the posterior inferior part of the cerebellum, and that artery is called as the posterior inferior artery. Posterior inferior cerebellar artery, which is a branch of vertebral artery, and here it joins to form the basilar artery. So here at the open medulla, here we have the basilar artery, which is formed by joining of the two vertebral artery, and before joining of the vertebral artery, each vertebral artery gives posterior inferior cerebellar artery as branch. So this posterior inferior cerebellar artery, so these are the branches which supplies the open medulla. If there is a occlusion here, then it results in lesion of this part of medulla, and it is called as medial medullary syndrome. So if there is occlusion of the medullary branch, so it gives the clinical syndrome, that is the medial medullary syndrome. If there is occlusion in this part, that is the posterior inferior cerebellar artery, so then it leads to lateral medullary syndrome. It leads to the lesion of this part, which is known as lateral medullary syndrome. So first, let us discuss the medial medullary syndrome. I am copying this part for better understanding. Okay, so first we will discuss about the medial medullary syndrome. So which branch occlusion? Medullary branches of vertebral artery. So what structures will be compromised? These medial structures will be compromised. So let us see which structures are being compromised. Number one, starting from posterior to anterior. So one is the hypoglossal nucleus and nerve. Hypoglossal nerve supplies the muscles of tongue, so there will be ipsilateral tongue motor paralysis. Number two, here is the medial lemniscus. So the medial lemniscus is lost. It has already crossed, so there will be contralateral loss of fine touch, proprioception, joint and vibration sensation. Now number three, corticospinal tract will be lost. So there will be contralateral hemiplegia. And lastly, number four, the arcuate nucleus is lost. So it will cause no such significant effect. So this was the medial medullary syndrome. Let us now see the lateral medullary syndrome. Number two, lateral medullary syndrome. Reason of lateral medullary syndrome is the posterior inferior cerebellar artery occlusion. So let us see. Here is our drawing. So which structures will be lost? These are the structures that will be lost. So let us discuss. Number one, here is the cranial nerve 9, 10, 11 at different level. Here is the cranial nerve 12. So number one, there is the posterior spinocerebellar tract that will lead to ipsilateral partial hypotonia, because spinocerebellar tract is responsible for maintaining the tone of the muscle. So there will, and it carries the ipsilateral, it supplies the ipsilateral portion. So it causes ipsilateral partial hypotonia. Next, anterior spinocerebellar tract. This will lead to contralateral partial hypotonia. Both of these will lead to bilateral muscle weakness. Next, number 3, nucleus ambiguous. Nucleus ambiguous will be lost. So this supplies the larynx, pharynx, muscles of larynx and pharynx. So this will lead to ipsilateral. This will lead to difficulty in speech and deglutination. Number 4, spinal lemniscus. Spinal lemniscus, which is the discuss of spinothalamic tract. This will lead to contralateral pain, temperature, pressure, touch, sensation, loss. Number 5, there is a spinal tract and nucleus of trigeminal. Spinal tract and nucleus of trigeminal. So this will lead to ipsilateral loss of pain, temperature, pressure, touch, sensation. And last but not the least, there will be damage to reticular formation, which controls the sympathetic activity of head neck. So there will be Horner's syndrome. If the sympathetic activity is compromised, so it will lead to ptosis, meiosis, and hydrosis, and etc. clinical features. So if we see the clinical feature, we can very well see. Suppose this is the person affected by lateral medullary syndrome. So whatever we discuss, there will be bilateral muscle weakness, there will be difficulty in speech and deglutination, there will be contralateral pain and temperature and pressure sensation loss of the body, and here it is the of head neck region. So in head neck region, suppose there is a loss of this. Suppose this is the right part and this is the left part. So suppose there is a loss of left part. So there will be the ipsilateral loss of head neck and contralateral loss of sensation on the rest of the part of the body. So this represent alternating hemianesthesia. Hemianesthesia. This was about the lateral medullary syndrome. So now let us discuss other brainstem lesion. So in the third type of brainstem lesion, that was the lateral and medial medullary, that was related to medulla. Now let us discuss about damage to pons. So here we have the syndrome is named as Miller-Gubler syndrome. Reason of this syndrome is the occlusion of the pontine branches of basilar artery. So let us see the pons. So here we have the pons. So let us see which pontine branches. So this will lead to the paralysis of this part. So let us see which structures are getting compromised. Number 1. Here is the nucleus of 6th and 7th cranial nerve. So nucleus of CN6. So this is the abducens nerve which supplies the lateral rectus muscle. So lateral rectus is responsible for keeping the eye laterally. So if this is compromised, so there will be medial squint. Ipsilateral. Second, there is loss of 7th nucleus of cranial nerve 7, which is the facial nerve. So there will be ipsilateral facial motor palsy. Number 3, we have the medial lemniscus. So there will be contralateral fine touch, proprioception, joint vibration sense loss. Number 4, we have the spinal lemniscus, which is the lemniscus of spinothalamic tract. So it will result in contralateral loss of pain, temperature, pressure, touch, sensation loss, and also corticospinal tract is also lost. So it will lead to contralateral hemiplegia. Now you may ask me some question that the corticopontine fibers are also getting lost, the anterior spinocerebellar tract is also getting lost, the spinal tract of trigeminal is also getting lost. So I want to say you that the corticopontine fibers are present abundantly, so these small loss will not lead to any major significant clinical feature. Again, same for the anterior spinocerebellar tract, because the other side is also present and there is present there, and another, the posterior spinocerebellar tract is also present, so there will be no significant involvement of this tract due to lesion. Next, next we will discuss damage to midbrain. So it has two syndromes. So the damage to the midbrain can occur at two regions. So here is a substantia nigra. So either there is lesion of this crus cerebri, or there is lesion at the tectum of the midbrain. Either there is lesion of the crus cerebri or there is the lesion at the tectum of the midbrain. Okay, so based on this, the lesion of the crus cerebri is called as Weber syndrome, and the lesion of the tectum is called as Benedict syndrome. So let us discuss these in details. So number one, we have the Weber syndrome. So what I have discussed, that it is lesion to the crus cerebri. Let me so here is a lesion to the crus cerebri. So what are the structures that will be damaged? Number one, third cranial nerve, which is the oculomotor nerve. It supplies the extraocular muscle, so there will be ipsilateral paralysis of extraocular muscle. So clinically there will be lateral squint because of unopposed action of lateral rectus muscle. Second thing, corticospinal tract, that will lead to contralateral hemiplegia. Here is the corticopontine. This will lead to no such significant effect. Next we have the Benedict syndrome. That is lesion to tegmentum. First, I will make you clear what is tectum, what is tegmentum, and what is crus cerebri. So here is the superior colliculus. Let us draw a line here. So this part is tectum, and this part is called as cerebral peduncle. Again, here we have the substantia nigra. This part of cerebral peduncle is called as tegmentum, and this part is called as crus cerebri. So tegmentum plus crus cerebri is equals to cerebral peduncle. So there is a lesion of the tegmentum here. Tegmentum and also some part of the tectum also. So what will be lost? Number one. So what structure will be lost? Number one. Let us go medial to lateral. So medial to laterally, red nucleus, that will lead to cerebellar lesion and motor weakness. Number two, medial lemniscus. Contralateral loss of fine touch proprioception, joint vibration sense. Third, trigeminal lemniscus, contralateral loss of all sensation of face, of head neck. Number four, spinal lemniscus, contralateral loss of pain, temperature, pressure, touch of full body. And number five, cranial nerve. Third, so there will be its lateral squint and the medial rectus. So this is in short various lesion and their effect of brainstem. So thank you for watching. If you have any doubt, you can ask me. Let's discuss about the cerebellum. So cerebellum is a part of hindbrain. Now the cerebellum can be divided into various parts by three classifications. One is the anatomic classification, which is based on the morphology of cerebellum. One is the phylogenetic classification, which is previously it was well accepted, but now the based on the function of cerebellum, well accepted is a phylogenetic classification. Now the physiological classification is well accepted as to determine the function of cerebellum. So let us first we will observe the what is the anatomical classification. So let us learn the anatomy first. First, let us draw the this is the mid brain stem. Here is the midbrain, pons and medulla, and behind the pons we have the cerebellum. I am drawing the so this is the fourth ventricle and the central canal. If we draw a cut section, that is sagittal section of the cerebellum, we can see various leaf-like white matters. This leaf-like presentation of white matter is called as folium, and the part of cortex that is intervening between two folium is called as fissure. Okay. Now, if we see the cross structure of the cerebellum, it has a long band of white fiber which is known as vermis, and this vermis connects two cerebellar hemisphere. So these are the cerebellar hemisphere, and the both two cerebellar hemisphere is connected by a tuft of nerve fiber, white fiber, that is the vermis. So vermis is basically white fiber. Now let us see what are the parts of cerebellar hemisphere and vermis. So prepare like this, that the way I am teaching. So here this part is the lingula. The vermis is divided in several parts. First here is the lingula, then toward the part, so this is called as central lobule, this is called as culmen. Similarly here also we have two parts, and here is the another part. So this terminal part is the nodule, which has two lateral extension named as flocculus or flocculus. So these are the flocculus. The two part above the nodule are pyramids and uvula, and in between these there are three parts of vermis, that is declive, folium, tuber. You can very well see that these names, that is folium, tumor, lingula, nodule, these are plant derived names because this structure, this sagittal section of the cerebellum looks like arbor vitae, which was compared to tree of life. That's why as there is tree, that is plant, so these names are also plant derived names. Okay, now let us draw the parts of cerebellar hemisphere. So here is one part which is called named as ala, which is connected to central lobule. Below this, here it is known as quadrangular lobule. Let me draw other parts of the cerebellar hemisphere. So this is the quadrangular lobule. Here is the lobular simplex. Here it is the superior semilunar lobule. Here is the inferior semilunar lobule. Here is the biventral, and here lastly here is the tonsil. We can very well see that here lies a fissure between quadrangular lobule and lobular simplex. Then here lies another fissure between the superior semilunar and inferior semilunar lobule, and here lies another fissure which lies between the tonsil and the flocculus. So same in this side also. So this is how a cerebellum looks like. So now this fissure is termed as primary fissure or fissura prima. This fissure is termed as horizontal fissure. It is not much well defined fissure. And here this fissure is known as flocculonodular fissure. So this fissure divides the anatomical lobes of cerebellum. So this whole of the lobe is termed as the anterior lobe, This whole of the lobe from fissura prima to flocculonodular fissure, we have the posterior lobe, and from flocculonodular fissure, this lobe is termed as flocculonodular lobe because it includes the flocculus and the nodule. Now you may ask me that why I have drawn the lingula in black and the nodule in black, why I have drawn the central lobule, ala, quadrangular lobule, culmen in this blue, because these color code represent the phylogenetic classification of cerebellum. So phylogenetically, cerebellum was divided into, in black color it is the RK cerebellum, RK or primitive cerebellum, In the blue color, it is divided into paleo-cerebellum, and in the red color, it is the newly discovered part of cerebellum, the newly understand part of cerebellum, that is the neocerebellum. So based on these, the lobes and the part of vermis differs. Okay, so you can very well now understand which part of vermis and which part of the cerebral hemisphere comes under this phylogenetic classification. So we have completed till now the anatomic classification, the phylogenetic classification. The physiologic classification will be taught in the physiology part. Let us see the histology or the internal structure of cerebellum. So here in this video we will not discuss in very details of the histology. It will be the internal circuitry and other all will be discussed in the physiology class. Here I will just point out what points you will write when cerebellum slide comes in your exam. So here you have to write first that it is divided in two types of layer. One is the cortex and another is the medulla. So here we have the cerebellar medulla, and these three layers, that is the molecular layer, Purkinje and granular layer, this comes under the cortical layer. So there is the cortex, which is a cellular layer containing the neural cells, and here is the medulla containing the nerve fibers. Now cerebellum cortex is divided in three layers. One is the molecular layer, Purkinje layer, and granular layer. Now in the Purkinje layer there is presence of Purkinje cell, in the granular layer there is presence of granule cell, and the molecular layer it consists of very fine types of cell. It consists mainly of myelinated and unmyelinated nerve fiber, and it does not contain any specific kind of cell. So that is why it contains only two types of cell, that is the basket cell and the stellate cell. So it contains many myelinated and unmyelinated nerve fiber. That is why it appears pale. Now one thing, just a summary. So if we consider these as the cerebellum, here is the lingula, here is the nodule, here is central lobule, culmen, here is the uvula and pyramis, and here we have declive, folium and tuber. So the vermis I have drawn. Next let us draw the cerebellar hemisphere. So here is the ala, here is the quadrangular lobule, then here is the lobular simplex, superior semilunar lobule, inferior semilunar lobule, biventral and tonsil, and here is the flocculus. So this is the anterior lobe, this whole is the posterior lobe, and this is the flocculonodular lobe. Posterior lobe, anterior lobe, and flocculonodular lobe. So this is the summary of the morphology of cerebellum. Okay, now let us discuss the cerebellum and its external connections. So here I have drawn the cerebrum. Here is the cerebrum. Then I have drawn the brainstem structures. So here I have the brainstem structures. So here I have the brainstem structures, the midbrain, pons and medulla, and here I have the section of spinal cord. So first I will discuss what are the functions or the main function of cerebellum. So the function of cerebellum has a great history. That this function of cerebellum was not discovered collectively. It was discovered through a long process of time. The scientists understand first the cerebellum. Okay, so the function of cerebellum include, first is the maintenance of equilibrium. So the portion of cerebellum which maintains this is the archicerebellum. So first, so equilibrium function was discovered first for the cerebellum. That's why it was termed as archicerebellum. And the physiological part which performs this action is the vestibulocerebellum. Controlling nucleus is the nucleus fastigius. Then maintenance of muscle tone. It is controlled by the paleocerebellum. In the physiological part, responsible is the spinocerebellum. The controlling nucleus is called as nucleus interpositus. This nucleus interpositus consists of two closely placed nucleus. One is the globose, another is the emboliform. So globose and emboliform nucleus. Then the third and the final main function of cerebellum is the maintenance of fine coordinated motor movements. So this part is controlled by neocerebellum. And physiologically, in physiological classification, it is called as cerebrocerebellum. The controlling nucleus is the nucleus dentacus or dentate nucleus. Now the cerebellum has three connection with the brainstem. With the midbrain and some part of pons, it is connected through superior cerebellar peduncle. With the pons, it is connected via middle cerebellar peduncle, and with the medulla it is connected via inferior cerebellar peduncle. So the cerebellum is connected by midbrain, or to midbrain and pons by superior cerebellar peduncle. By the pons itself it is connected by the middle cerebellar peduncle, and by the medulla it is connected by the inferior cerebellar peduncle. Let us I draw the three parts of the cerebellum. So here I have the archicerebellum, here I have the paleocerebellum, and here I have the archicerebellum. Same thing I will draw here, archicerebellum, then paleocerebellum, then neocerebellum. Now let us draw the nucleus. In the archicerebellum part, there is the nucleus fastigius. In the paliocerebellum, there is the nucleus interpositus, and in the neocerebellum, here there is the nucleus denticus. Now we will describe how the various functions are mediated via external connection of cerebellum. So first function was the maintenance of equilibrium. So maintenance of equilibrium is via the semicircular canals and the vestibule of the inner ear. So here is the semicircular canal and the vestibule. So here is the inner ear, which controls the which contains the receptor for the equilibrium. So it is carried out. The information is carried out to the vestibular nucleus by 8th cranial nerve, the vestibulocochlear nerve, to the medulla. From there, the information is taken by the inferior cerebellar peduncle to the nucleus fastigius. From nucleus fastigius, in the nucleus fastigius, the information is processed, and final information for maintenance of equilibrium is sent to the reticular formation. So here, the brainstem consists of the reticular formation. From there, reticulospinal tract takes the information and relays on the designated lower motor neuron to supply the muscle needed for maintenance of equilibrium. So let us summarize. So from the inner ear, equilibrium sensation is received. It is carried towards the vestibular nucleus present in the medulla via vestibulocochlear nerve. From the vestibular nucleus, the information is carried to the nucleus fastigius of archicerebellum via inferior cerebellar peduncle, and from the nucleus fastigius, that information is checked, and it then stimulates the reticular formation. From there, the reticulospinal tract relays over the lower motor neuron and do the necessary adjustments by supplying the muscle responsible for postural changes. Okay, so maintenance of equilibrium is done. Now maintenance of muscle tone. So here we have the receptor of muscle tone. It is the muscle spindle. From the muscle spindle, the information is carried through by the posterior and anterior spinocerebellar tracts. The fibers of posterior spinocerebellar tract ascends up at the level of medulla. It passes through the inferior cerebellar peduncle and carries the information to the nucleus interpositus. Whereas the fibers of anterior spinocerebellar tract ascends up, then it enters the cerebellum, then it crosses the midline and enters the cerebellum via superior cerebellar peduncle, to relay the information over the nucleus interpositus. Then the information from the nucleus interpositus is then sent to the reticular formation via the middle cerebellar peduncle, and the reticulospinal tract do the necessary adjustments. So here is the anterior spinocerebellar tract, which carries contralateral information, and here is the posterior spinocerebellar tract, which conveys the ipsilateral information. So the muscle spindle is sent some change in the muscle tone. That information is carried by the sensory nerve to the posterior spinocerebellar tract, and some sensation is carried out by the anterior spinocerebellar tract. So the posterior spinocerebellar tract ascends and it enters the cerebellum via inferior cerebellar peduncle to relay over the nucleus interpositus. From nucleus interpositus, the signal is sent to the reticular formation, and the reticulospinal tract supplies the lower motor neuron to do the necessary adjustment in the muscle tone. Now similarly, the anterior spinocerebellar tract, it carries the information from the contralateral side, then it ascends up, then it ascends up to the midbrain, and it then crosses the midline and enters the cerebellum via superior cerebellar peduncle to relay over the nucleus interpositus, and then from the nucleus interpositus, information is carried to the reticular formation, and the reticulospinal tract do the necessary adjustment in the muscle tone by relaying over the lower motor neuron. Okay, so maintenance of muscle tone is also done. Now let us see how fine coordinated motor movements are maintained. So from the brain, high voltage information is carried by the corticopontine fiber, and it descends through the pons, where it is captured by the pontine nucleus, and then this information is sent to the nucleus denticus, or the dentate nucleus, by crossing the midline. From the nucleus denticus, this information is tuned and refined and sent back to the red nucleus present in the midbrain. From the red nucleus, the information is sent to the thalamus, and from the thalamus it is sent again back to the cerebral cortex. So from cerebral cortex now tuned information is carried out by the corticospinal tract to its designated destination. So this pathway is known as cortico-ponto-cerebello-rubral pathway. So here we have the red nucleus. So this is the red nucleus. Okay, now one thing, that the high voltage signal is carried out by the corticopontine fiber, then it is caught. Now many of the fibers are caught by the pontine nucleus present at the pons, and that information is taken via the middle cerebral peduncle to the dentate nucleus. Some fibers does not relay on this nucleus and descends down up to the arcuate nucleus present in the medulla. So here is the arcuate nucleus which is present in the medulla. From this, via external and internal arcuate fiber, the information is taken to the inferior cerebellar peduncle, then to the neocerebellum, and then to the nucleus denticus. And again, via the cerebello-rubral tract, this information is carried to the red nucleus, and from the rubro-thalamic tract, it is conveyed to the thalamus, and from the thalamic radiation, it is then goes to the cortex, and this forms the coordination of the movement very fine, very fine. So, else, if this pathway is lost, there will be error in the fine coordinated movement. So let us again discuss all the pathway. So for equilibrium, so pathway number 1 for equilibrium change sensed by vestibule of inner ear, cranial nerve 8, that is the vestibular cochlear nerve, vestibular nuclei present in the medulla, then via inferior cerebellar peduncle it goes to nucleus fastigius, which gives information to reticular formation via middle cerebellar peduncle, which gives information to reticulospinal tract, which gives supply to lower motor neuron for muscle related to postural control. Equilibrium maintained. Now second, muscle tone sensed any change sensed by the receptor in muscle, which is the muscle spindle, then it is carried by ipsilaterally posterior spinocerebellar tract, ফার্স্ট হচ্ছে সেন্সারি নার্ভ টু স্পাইনাল কর্ড, দেন এখানে দুটোভাবে ডিভাইড হয়ে যাচ্ছে। ওয়ান ইজ দ্য ইপসিল্যাটারাল পোস্টিরিয়ার স্পাইনো সেরিবেলার ট্র্যাক্ট, দেন ইজ দ্য কন্ট্রাল্যাটারাল এন্টিরিয়ার স্পাইনো cerebellar tract. This information is carried through the inferior cerebellar peduncle, then to the nucleus interpositus, then anterior spinocerebellar tract ascends up to midbrain and upper pons, superior cerebellar peduncle, nucleus interpositus, reticular formation, spinal tract, and lower motor neuron. Next, maintenance of fine coordinated muscle movement. So for that, high voltage signal from motor cortex, corticopontine fiber makes relay on pontine nucleus. From that, middle cerebellar peduncle, nucleus denticus, modulation of signal. Then হচ্ছে cerebello-rubral tract, red nucleus, fine tune info reach the cerebral cortex, then it is sent to corticospinal tract, then fine muscle movement. Some descend down, relay on arcuate nucleus in medulla, internal and external arcuate fiber, inferior cerebellar peduncle, and the nucleus dentatus. So this is all regarding the external connection of cerebellum. So lastly, let us discuss about the clinical feature of cerebellar lesion. So for this, remember a mnemonic that is vanished, NISHD, that is vertigo, ataxia, nystagmus, I for intentional tremor, S for slurred speech, H for hypotonia, which is typical of cerebellar lesion, then E for exaggerated broad gait because of loss of equilibrium, then D for dysdiadochokinesia, because of loss of fine coordinated movement. Nystagmus is involuntary movement of eyeball. About the cerebral circulation. So the cerebral circulation include both arterial supply and venous drainage. Venous drainage will be discussed along with the dural venous sinus topic in head neck. Now a small introduction regarding cerebral circulation. So the cerebral circulation receives 20% of cardiac output and it utilizes 25% of total oxygen, that is the oxygen demand is 25%, and 750 ml of blood circulates per minute. Now coming to the arterial supply of the brain. So there it is by the circle of Willis. It has various other names, that is circulus arteriosus and polygon of Willis. So the circle of Willis, this name was coined by Thomas Willis, who was an English physician. So basically the circle of Willis is an anastomotic channel which supplies various parts of the brain. Now let us first see how it is formed, the circle of Willis. So first there are two arterial systems present in the circle of Willis. One is the carotid and one is the vertebral arterial system. I will start along with the vertebral arterial system. So the two vertebral arteries entering into the cranial cavity approaches each other and joins to form the basilar artery. Before joining, it gives two branches. One is the posterior inferior cerebellar artery and the anterior spinal artery. After forming the basilar artery, which is this artery, the basilar artery gives several branches. One is, here is the anterior inferior cerebellar artery, labyrinthine artery or artery to the towards the internal ear, pontine branches. There are several pontine branches. Finally, superior cerebellar artery, and the basilar artery terminates by dividing into posterior cerebral artery, two posterior cerebral artery, left and right posterior cerebral artery. Now coming to the carotid system, arterial system. So the carotid arterial system is formed by the internal carotid artery which enters the cranial cavity. It has several parts. It will be discussed in the head neck question. So it enters through the carotid canal and forms the circle of Willis by giving its anterior cerebral arterial branches. It gives one anterior cerebral artery. The left internal carotid artery gives the left-sided anterior cerebral artery branch, and the right internal carotid artery gives the right anterior cerebral artery branch. The two anterior cerebral artery is joined together by the anterior communicating artery. The internal carotid artery again gives another branch to join the posterior cerebral artery of the respective sides, which is also known as the posterior communicating artery. And the internal carotid artery terminates by giving the branch anterior cerebral artery, and itself continues as the middle cerebral artery. So let us write the names. So this is the anterior cerebral artery, this is the middle cerebral artery, this is the internal carotid artery, which terminates by giving main two branches, that is the anterior cerebral artery and middle cerebral artery, and the posterior communicating artery. Remember, posterior communicating artery is the branch of internal carotid artery. Here it is the posterior cerebral artery, here it is the superior cerebellar artery, which is a branch of basilar artery. These are the pontine branches of basilar artery. This is the labyrinthine artery. This is anterior inferior cerebellar artery, which is the first branch of basilar artery, and this is the vertebral artery, which forms the basilar artery, and this is the posterior inferior cerebellar artery, and this is the anterior spinal artery. It also forms the vertebral artery also gives the posterior spinal artery also, and here it is the anterior communicating artery. So this is the diagram of circle of Willis. You must need to draw this. So let us again write which arteries forms the circle of Willis. So the circle of Willis is formed by 2 internal carotid artery, 2 anterior cerebral artery, 2 posterior cerebral artery, 2 posterior communicating artery, and 1 anterior communicating artery. So 2 anterior cerebral artery, 2 internal carotid artery, 2 posterior communicating artery, 2 posterior cerebral artery, and 1 anterior communicating artery. So this is how the circle of Willis is formed. Remember, the middle cerebral artery does not form the circle of Willis. Now, what is the function of circle of Willis? So it equalizes the blood flow. It equalizes the blood flow. That is, it acts as a homeostatic anastomotic channel. That is, if some of the area of brain is getting less blood supply, then it does some homeostatic adjustment and restore the blood flow to that area of brain and equalizes the blood flow. So it equalizes the blood flow. Remember, there is very little intermixing of the blood take place. That means the blood from the right side internal carotid artery and the right sided vertebral artery, there is very little intermixing between the right and left side. However, it acts as a principal collateral channel. That is, if there is any occlusion of one arterial system, then the anastomotic channels will open and it will equalize the blood flow and prevent compromisation of the blood flow to the brain. If there is obstruction of internal carotid artery, then blood flow from external carotid artery will flow through anastomosis in face and scalp. Now, there are some of the anomalies you need to know. So, the anomalies that might be found in the circle of Willis are the posterior cerebral artery arising from internal carotid artery, which is now the posterior cerebral artery normally arises from the basilar artery, but it may arise from internal carotid artery. The ACOM may be double, that is the anterior communicating artery may be double, and the PCOM may be absent unilaterally or bilaterally. Now, coming to the branches of the circle of Willis. So, there are 3 types of branches of circle of Willis. One is the cortical branch, second is the central end arteries, and third is the perforating arteries. The cortical branches supply the cortex, and this is one of the most important arteries which are clinically very relevant to us. Then we have the central end arteries which supplies various internal brain structures like basal ganglia, thalamus, hypothalamus. So these are supplied by the central end arteries. And the perforating arteries. Perforating arteries, these are various numerous small vessels that penetrate the brain substance through anterior and posterior perforated substance. We will discuss this later. So let us see how these branches are situated in the circle of Willis. Let us again draw the circle of Willis. So here is the basilar artery which divides into 2 posterior cerebral artery. Here is the posterior communicating artery. Here is the internal carotid artery, which again gives middle cerebral artery, posterior communicating artery, and the anterior cerebral artery as branches. Here is the anterior communicating artery. So this is a very schematic diagram. So now I am drawing the branches. So first is the anteromedial branch. So anteromedial branch arises from the anterior communicating artery and some part of the anterior cerebral artery. So this is the anterior medial branch, which supplies the anterior hypothalamus. Then let us see where are the anterolateral branch. So anterolateral branch arises from the middle cerebral artery. So these are the anterolateral branch which arise from the middle cerebral artery, which gives supplies to the striatum and internal capsule. Then we have the posteromedial branch. The posteromedial branches are arising from the posterior cerebral artery. So it supplies the posterior hypothalamus and the subthalamus and the anteromedial thalamus. Then we have the posterolateral arteries. So here we have the posterolateral arteries. Here we have the posterolateral arteries which supply the caudal thalamus. Okay, so this was in short anteromedial arteries, anterolateral arteries, posteromedial and posterolateral. Now let us discuss about the perforating arteries. So the perforating arteries are those arteries which perforate the brain substance to supply the internal structures of brain. So let us suppose this is our brain, and here is the main artery present. So and here are some of the internal brain structures. Let us suppose this is the basal ganglia. So the perforating branches pierces the brain substance and supply these structures. The point where it pierces or the region from where it pierces are called as perforating substance. So these areas are called as perforating substance. So and these are the perforating branch. So I have made a table to summarize this. So that there are 2 types of perforating arteries. One is the anterior perforating and the posterior perforating artery. The anterior perforating artery, it arises from the anterior communicating artery, anterior cerebral artery, and the middle cerebral artery. That is from the anterior circulation of circle of Willis. The posterior perforating artery, it arises from the posterior cerebral and the posterior communicating artery. How it enters the brain? The anterior perforating artery enters the brain to anterior perforated substance, which lies behind the olfactory trigone and in front of the optic tract. You can see the diagram in the interpeduncular fossa. It is discussed there. And the posterior perforating arteries, these enters through the brain through the posterior perforating substance. Now supply the anterior perforating artery supplies the basal ganglia, optic chiasma, internal capsule, and the hypothalamus, and the posterior perforating artery supplies the ventral portion of the midbrain and subthalamus and hypothalamus. Now let us discuss the most important topic of this most important part of this topic, that is the cerebral arterial supply, that is domain of supply by anterior cerebral artery, posterior cerebral artery, and middle cerebral artery. So first, you can jolly well see what are the distribution or the area of supply of the anterior, middle, and posterior cerebral artery. For the anterior cerebral artery, the region that is supplied is mainly the medial surface. Among the medial surface and the orbital surface. Among the medial surface of frontal and the parietal lobe, this part is supplied by the anterior cerebral artery. So area of supply is the orbital and medial surface of frontal and parietal lobe, and a narrow portion of the superolateral surface, So these are the parts which are supplied by the anterior cerebral artery. Now the most important part here is the effect of lesion. So as it supplies the orbital and medial surface of frontal and parietal lobe, so if there is some lesion, then there will be difficulty in prefrontal lobe function, that is cognitive thinking, judgment, motor initiation, self monitoring. And it also supplies the narrow part over the superolateral surface. According to the motor homunculus, the narrow part of the superolateral surface there is the representation of the distal limb structures. So there will be motor disturbance in contralateral distal limb structures, that is the lower limb structures. Then coming to the middle cerebral artery occupies the entire superolateral surface, that is, it supplies the whole of the somatosensory cortex, the motor cortex, the language area which is present here, then the auditory area which is present here, which is present in the temporal lobe. So somatosensory area, if there is some lesion, there will be contralateral sensory loss of face, arms, hand, more than that of legs. Same for motor cortex, there will be contralateral motor loss if there is some lesion. And for the language area, there will be aphasia if there is some lesion. In case of Broca's, there will be decreased production of the speech, that means there will be motor speech aphasia, and if Wernicke's area is lost, then there will be comprehension loss or sensory speech aphasia. If there is some lesion at the auditory area, then there will be auditory sensation loss. Now coming to the posterior cerebral artery, the area that it supplies is the inferior and the anterior part of the temporal lobe and the inferior and medial occipital lobes, that is the visual area. So the temporal lobe is associated with the memory generation, so there will be memory impairment, and if the visual area is compromised, so there will be visual disturbance and there will be loss of field of vision, which is also called as hemianopia. There will be half of loss of vision. Now let us see what are the branches of anterior cerebral, middle cerebral and posterior cerebral artery. For this we will draw some diagrams. So here I am drawing the corpus callosum, and in the brain, there is the brain. For the middle cerebral artery, we will draw the superolateral surface. For the posterior cerebral artery, we will again take the medial surface. So this is the medial surface which we are viewing, and this is the superolateral surface of the brain. So let us see how the anterior cerebral artery gives the branches. So the anterior cerebral artery, after emerging from the circle of Willis, it ascends upward and also moves posteriorly along the pericallosal sulcus. In this course, it gives some branches. Anteriorly, it gives the orbital branch. Then again, it gives the branch to the frontal pole, so it is called as frontopolar branch. It gives a branch which is the calloso-marginal branch, and it gives another branch which runs along the pericallosal sulcus. And this pericallosal branch supplies or gives supply to the corpus callosum. So this is the area of supply for the anterior cerebral artery. So there is the orbital branch, then there is the frontopolar branch, then there is the calloso-marginal branch, and there is the pericallosal branch. Now coming to the middle cerebral artery. So the middle cerebral artery, after its formation, it winds around the lateral sulcus and occupies the lateral sulcus and moves posteriorly along the lateral sulcus up to the posterior rami, where it ends by giving a branch which is named as the angular branch. During its course, it gives the several branches. Anteriorly, through the anterior rami or the ascending rami, it gives the orbitofrontal branch, then the pre-Rolandic branch, then the Rolandic branch, then the anterior and posterior parietal branch, and it also gives the anterior temporal and posterior temporal branch. So this is the area of supply of the middle cerebral artery. So let us write the branches. So this is the orbitofrontal branch. These two are the anterior and posterior temporal branch. This is the pre-Rolandic branch. This is the Rolandic branch. These two are the anterior and posterior parietal branch, and this is the last is the angular branch. Now let us see the posterior cerebral artery. Now the posterior cerebral artery, it has two branches. One is the temporal branch and another is the occipital branch or the internal occipital branch. So the posterior cerebral artery runs posteriorly, and first it gives a temporal branch, then it divides near the calcarine sulcus into parieto-occipital branch and the calcarine branch. So this is the area of supply of the posterior cerebral artery. So first it gives the temporal branch, then the posterior cerebral artery terminates by dividing into a parieto-occipital branch and calcarine branch. The parieto-occipital branch occupies the parieto-occipital sulcus and the calcarine branch occupies the calcarine sulcus. So this is all regarding the arterial supply of brain. I hope this has helped you a lot, and I hope that you understand how to write during exam. So thank you, and stay tuned for another lectures.